Are you able to share screen at all?
Let me see. No, I'm not able to do anything.
All right, let me try again here.
Thank you.
Yeah, I can't control any of that. Oh, wrong one.
It says all panelists.
Let me see.
This is the right link, right?
Mm-hmm, yeah.
Yeah.
This is it's a webinar type Zoom. So
Oh, I see. Okay.
we're trying it for the first time. Speaker view, gallery. Let me see.
You can hear me all right, though, right?
Yes.
Who can share? All panelists, all panelists. Okay, those are correct.
Can you share, Gina?
Yeah,
I know I made you a host.
Yeah, you made me a host, but I see that the check marks are for advanced sharing options. They both say
Yeah.
panelists.
And Arvind, you still don't see share screen for yourself at the bottom?
No.
Close that.
Can you make me a host for a little bit?
Yeah, let me see if I can do that.
That might give me video and.
You should be able to under participants.
Thank you.
And then more, maybe not.
Promote to panelists. There we go. I think that works.
Yay!
Okay, there we go.
Okay.
You need to enable my video as well, I think.
Wow.
FaceTime, HD. Okay.
I'm just going to make host.
There we go.
Okay. There we go.
Okay.
You have the power.
Right.
Nice seeing you guys. Thanks for having me today.
Thank you for coming.
For coming and being able to do this. I listened to part of your talk when you did the School of Medicine, the biomedical basics for the science writers boot camps. So I was able to watch some of that and it was really good.
Oh, thank you.
I did. And then I was talking with me. I'm like, oh, we should have him come speak with us then.
Did you hear us talking when you were waiting, Arvind?
Just for like maybe 2 seconds or something, 2 or 3 seconds.
It's weird that there's no waiting room.
Yeah, I was gonna say I'm kind of the waiting room last time.
Yeah.
I just heard voices. Is today's lecture mostly for students or is it like open to?
We open it to anybody who really wants to come. So you know how it is with faculty. It also depends on how their schedule's looking. But so we get a good mix of different students and faculty and like people coming in later when they have their free time. But we're also, again, we're going to record it so that we can put it back on the website. So anybody who can't attend today or, that they have to leave early, it's like, it's there.
Sure.
Gina, can you see this Q&A on the bottom? I'm trying to make sure we have
no open questions.
Yeah. But then I don't see it a way for me to submit a question either.
Are you guys at campus or at home?
At home.
I'm at home. This is totally a virtual background on for me. We're all at home. Although I live actually right by the Homewood campus. So if I needed to rush into the office for something, if I forgot it, I'm right there. Yeah, Camille, I don't see how somebody could submit a question.
I'm looking at the directions.
I can share my screen.
There's a raise hand feature. Oh, maybe we do the raise hand with participants. Let me click on participants. No. Okay. Yeah, maybe we just do the raise hand feature.
I can't see the raise hand feature on my end.
Oh, click on participants and then click on raise hand. Yeah.
Oh, I see it.
Because this Q&A is not making sense.
Can you guys see my screen?
We can.
Okay. What I'm going to do is I'm going to turn off my video while I'm talking just because I have some videos in there and I don't want the bandwidth to be hogged by the camera.
Okay.
just so that it runs. Can you see my slides changing as well?
Oh, yes.
Okay, cool. Great.
Excellent.
Interesting. All my controls disappear when I share screen.
Okay, Eugenia can hear us, so this is just how it's set up.
All right.
Yeah.
Oops. What did I do?
And we'll wait a few more minutes for more attendees and then we can get started.
Okay.
No, that's fine.
I feel like it should integrate elevator music or something like that. Waiting period. Camille, will you control the record feature?
Yes,
Like,
I can do that.
and then when it ends. Thanks. Oops.
So Gina, I might have to ask you to do a few things because I'm not a host or co-host anymore.
What do you
Since
mean?
I made Arvind host. Maybe during the Q&A.
All right.
Okay. We'll see what happens. We'll wait a couple more minutes.
So Gina, are you officially at the INBT or the communications office?
No, I'm officially with INBT.
Okay.
Yeah, so I strictly do like the INBT communications. I also help with within the INBT. There is the Physical Sciences Oncology Center, so I do the communications for them as well. No, I'm not in I'm not in the central office.
Okay. What do you think, Gina? Can we go ahead and get started?
We can go ahead and get started. And I'm doing the intro. So, all right. Well, welcome everybody. Good afternoon. If you don't know me, my name is Gina Wattis, and I'm the science writer for the Institute for NanoBioTechnology. We really appreciate you joining us today. Just to give you a heads up, we are recording this seminar. So if you're not able to attend the full session, or if you know someone who wanted to attend and it just didn't work out for them time-wise, I will be posting it on the INBT website definitely by next week. So you can go back there and check it out. We'll have it available for you. We'll also have time at the end for a Q&A session. So hopefully you can stay till then, and Camille Mathis will also help facilitate that, hopefully, as long as Zoom works well. So let's go ahead and get started. And I'm so happy to introduce our guest. We have Arvind Pathak, and he has a Bachelor's of Science in Industrial Electronics and a PhD in Functional Imaging. He completed his postdoc work in molecular imaging at the Johns Hopkins University School of Medicine. He is currently an associate professor in radiology, oncology, biomedical, and electrical engineering here at Hopkins. He's also a member of the Sidney Kimmel Comprehensive Cancer Center, the Institute for Computational Medicine, and of course, a member of the INBT here. So Arvind, it's your show now, and you can take it away. And thank you again for joining us.
Thank you, Gina and Camille, for having me. It's a real delight to be with everyone. Good afternoon, everybody. I'm just going to share my slides real quick and turn off my video to make this easier. Okay, so like Gina said, I'm in the Department of Radiology with appointments in biomedical and electrical engineering. And what I thought I'd do over the next 35 to 40 minutes is showcase some of the work that's underway in our lab, and showcase some of the ways in which we can leverage a variety of imaging tools to characterize the vasculature or blood vessels in a range of applications, ranging from cancer to tissue engineering to neuroscience. So that's the link to our lab's website. So if you have any questions regarding some of the work I'm presenting or any of the protocols I present, please feel free to go there. It's a slightly dated website, but a lot of key information is still available there. So our lab develops new imaging methods, computational methods, and visualization tools to make visible critical aspects of cancer, neurobiology, and tissue engineering. We work in 4 large domains. One is the generation of image-based biomarkers. We develop image-based systems and computational biology tools. We're one of the few labs in the world that does image-based tissue engineering. And I'll show you examples of how we also do image-based neuroengineering. On the bottom are some of the journal covers that our lab's work has been featured on. So in our lab, we have employed what's called an image-based paradigm to look at disease or answer really critical questions about the underlying biology. So this graph here, or schematic, kind of shows us what I mean by this image-based paradigm. The idea is to gain new insights into the underlying biology, and we do that in cancer, neuroscience, and tissue engineering domains. And the idea is to be able to answer new biological questions. And the way we do that is by developing or harnessing technology. By technology, I mean multi-modality imaging methods, multi-scale imaging methods, and even multi-dimensional imaging methods. And then what we do is we generate new data from these imaging methods that we incorporate in different computational tools. These tools could range from the development of new software, models, visualization approaches, atlases, and data repositories. And then we can use those to develop new hypotheses, and the entire loop kind of like feeds in on itself. And as you can see, this involves many different areas of expertise. So our lab is very interdisciplinary in nature. So we have people all the way from the School of Medicine to the School of Engineering, the School of Public Health, and so on. So I encourage you to visit our lab's website. So what actually drives the advances in this area of image-based systems biology or image-based engineering? So there are 4 key areas. One is the development and availability of new contrast agents and imaging methods. The second is the availability of cheaper and more powerful computational tools. The second is the rise of data visualization toolkits, as well as design thinking approaches. And finally, of course, the availability of components to create novel instrumentation systems. So our lab kind of straddles these 4 areas, and the intersection of these 4 areas is what allows us to make inroads in these 3 research domains that we are working in. So one of the primary focuses of our lab is to look at the role of blood vessels in disease and in health. And the reason why this is important is almost every pathology that we investigate involves some kind of abnormal blood vessel architecture. And when you have abnormal blood vessels, whether it's in a tumor or a tissue like the brain, for example, in stroke, what you get is abnormal hemodynamics. So the blood flow in those blood vessels tends to be abnormal. Then the mechanical properties of the blood itself tend to be abnormal. And then finally, around the blood vessels, we have an abnormal interstitial space. And then downstream of that, what we get is what's called an abnormal tissue or tumor microenvironment, where because the blood vessels and the blood flow is so abnormal, oxygenation, pH, the delivery of nutrients, clearance of waste products, and even metabolism in those organ systems tends to be abnormal. And then further downstream of that, we have a whole variety of cellular and molecular events or effects that are all consequences of an abnormal vessel architecture. So our lab is focused on characterizing these abnormal blood vessels. When we characterize the differences between healthy and abnormal blood vessels structurally, it's known as structural phenotyping. We can also characterize as differences in their function, which is known as functional phenotyping. And then we can characterize the effect that they have on the entire tissue microenvironment, which is known as microenvironmental phenotyping. So what I'm going to start with is an example from cancer. So the whole idea is to get a better handle on what happens inside a breast tumor, for example, when you have an abnormal vessel structure. So the goal was to actually model tumor angiogenesis, or the formation of new blood vessels in tumors. And the way we propose to do that is by obtaining the tissue or vascular geometry using some kind of 3D imaging method. Once you know the vascular geometry, we can actually set up mathematical models that predict the blood flow in those vessels. Once we know the blood flow, we can predict what the oxygen transport is going to be in the tissue. And from that, we can predict what the secretion of different proteins and growth factors is going to be. And then the microenvironmental cues that these proteins and growth factors produce actually induce blood vessel formation, and the entire loop kind of feeds back on itself. So this is a very well-established pipeline in the systems biology literature. However, the one big difference between what we do and what most labs do is that this entire pipeline is informed by imaging data. So our tool of choice, rather than molecular biology tools or microscopy tools, are 3D imaging data. So we use 3D imaging to obtain the vascular geometry, and use imaging data to validate the prediction of these models as well. So people have traditionally used very simple images to do this in the past. So this is just a photomicrograph of a tissue bed labeled or injected with India ink, and you can see the blood vessels light up very nicely. But the problem with this is that it's a very small area from a 2D image. People have also looked at different kind of mathematical approaches where they've approximated the blood vessel structure using finite element approaches. So that's shown here. With the advent of more complicated microscopy tools like multiphoton and laser scanning microscopes, we can actually obtain 3D information about the vessel structure, like shown here. But again, the problem with all these methods is that it doesn't give us an appreciation of the vasculature in an entire organ or an entire tumor. So some time ago, we use a combination of high-resolution MRI and high-resolution micro-CT to create images that look like this. So this is a human tumor grown inside a mouse. The red are the blood vessels in the tumor. So there are about 100,000 blood vessel segments in this tumor, color-coded by their average radius. And then the gray is the soft tissue contrast obtained from MRI. So we combine images from these different imaging modalities to create atlases of these tumors, if you will. And then once we have the 3D structure of the tumor, we can actually set up really sophisticated mathematical models to simulate the blood flow in these tumors. And this is just a schematic from a paper we published a few years ago where we simulated the whole tumor blood flow. And this is, again, not something new. The new part of this study is the fact that we use imaging data on which to predicate these models. So once you have the vessel structure, we can set up Poiseuille's law for the pressure difference in the vessel segments. We can set up the rheological equations, which are the mechanical properties of the blood. We can set up the boundary conditions, the model for the plasma exchange, and then we can use nonlinear optimization approaches to solve for these pressure boundary conditions. And then we can simulate the oxygen flux, calculate the oxygen tension, and finally the blood flow. And this is work done by Dr. Stamatelos in Dr. Sasha Popol's lab, who's a longtime collaborator. And it was featured on the cover of Microvascular Research because, to the best of our knowledge, this was the world's first simulation of blood flow in an entire tumor as opposed to just a small focused or just some segment or region of interest of a tumor. So this is very exciting. But in cancer, especially in breast cancer, a big problem has been the issue of cancer heterogeneity. And what we mean by this is, let's say a woman presents in the clinic with a kind of breast tumor. Depending upon where her tumor is, tumor biopsy sampled from, the physician can actually get very different results because the cells are different in different parts of the tumor. So the idea is to somehow get an appreciation and a handle on what is known as cancer heterogeneity using an array of tools in the preclinical space. So what I mean by heterogeneity in cancer is basically differences that can arise at multiple levels. For example, we're all familiar with interpatient heterogeneity. So one patient's breast tumor is very different from another's. We have something called intrapatient heterogeneity, where depending upon the site in the body, you'll get differences in the cancer cells. So for example, a breast cancer is very different from cancer that has spread to the spine or the lungs. We get heterogeneity at the level of the individual tumor. So like I said, depending upon where we sample the tumor, we get very different characteristics. And then finally, of course, we have heterogeneity at the genetic level. So the question is, how do we investigate these phenomena and do it in a meaningful way? We cannot do this in patients, especially very sick patients, because we cannot bring them in for repeated biopsies. It's a very invasive procedure and uncomfortable. It's also very difficult to do large cohorts of animal studies because they're expensive, and then animal studies have their own issues, so it's hard to do 200-300 animals. So what we do is we tend to turn to what I call the in silico space, or basically the modeling or computational biology space. And the idea is to develop new tools to elucidate tumor heterogeneity using a combination of imaging and modeling. So what we did is, just like before, we acquired images from human breast cancers implanted in animals, and then we created an entire library of tumors using high-resolution 3D imaging. And to simulate breast cancer at an early and late stage, what we did was we acquired samples at what I call post-inoculation week 3, which mimics early-stage breast cancer. And then we obtained a bunch of other samples from post-inoculation week 5, which mimics more advanced stage breast cancer. And then what we did was we combined images that we obtained from MRI, CT, and optics to basically create a multi-layered atlas of a breast cancer model, if you will. And then Akanksha Bhargava, who's a fellow in my lab, she decided to actually characterize the structural and functional heterogeneity in these tumors in terms of the physical as well as emergent parameters. So she looked at all the different morphological variables that one can get from the underlying vessel structure. So this ranges from simple parameters like vessel diameter all the way to things like surface area density and the distance to the nearest vessel. And then once we have the 3D vessel structure, we can actually calculate or simulate different functional variables. So we can actually compute what the blood flow is going to be, what the oxygenation is going to be, what the hematocrit is, and so on and so forth. And these are extremely important parameters in terms of the underlying tumor biology or physiology, because they inform us as to what the tumor microenvironment is doing. And if we delivered a drug, for example, would it get distributed all throughout the tumor? We can actually predict that based upon what we know about tumor blood flow. So like I said, we're a 3D imaging lab that also leverages a whole array of visualization tools as well. So shown here is data that Akanksha has computed for another 3D tumor, where all the blood vessels are labeled in blue. And then blood vessels which have very low flows and very low oxygenations are kind of labeled in white. So this is a 3D image that comprises of close to 100,000 different vessel segments. Once we know the vessel structure, we can not just calculate the flow, but we can actually determine how tortuous the vessels are. That means how far does the vessel geometry depart from normal. We can make maps of the blood flow velocity in these tumors, so you can actually see these pockets of high velocity flow. We can look at the oxygenation, so we can actually see areas where the oxygenation is relatively high, but most of the tumor tends to be hypoxic or low oxygenated or poorly oxygenated. And this is well known because the blood flow in these tumors tends to be very poor. And then you can actually combine all these data to identify the emergent properties of the tumor microenvironment or the tumor. So for example, we can actually identify areas that are very poorly perfused and hypoxic by combining the velocity data with the oxygenation data. We can identify areas that have medium to low perfusion. We can identify areas that are kind of very high in terms of their perfusion velocities and oxygenation. And then we have very unique vessel segments where they are dilated and very low resistance vessels. So again, this is a very powerful tool where we can now use imaging data to create sophisticated mathematical models and then simulate the emergent properties of a tumor and identify these unique vascular niches within the tumor. Another thing that we do in the lab is, of course, develop our own imaging tools and methods. So like I said, abnormal hemodynamics result in an abnormal tumor microenvironment. And MRI and PET often lack the resolution to look at individual blood vessels. So it's very hard to look at what happens in an early stage of a tumor in terms of blood flow changes. So we resort to things like optical imaging methods, where we can actually get microvascular resolution data that's dynamic in nature. And I'm going to show you examples of that. And this is not something unique to our lab. There's a lot of labs all over the world, like the Dewhurst lab and Jain lab, etc., which have leveraged these optical imaging methods in very elegant ways to study tumor hemodynamics. But the problem with most of these approaches is that they extract summary hemodynamic parameters, and they don't look at actually every single blood vessel to look at and get a handle on what I alluded to earlier, which is basically tumor heterogeneity. So we created a new toolbox to explore the temporal relationships in tumor hemodynamics at their native and microvascular spatial scales. So most optical imaging methods, and a lot of you will be familiar with this from your experiences with common microscopes, whether it's multiphoton or laser scanning microscope, or even just simple epifluorescence microscopy, we often look at a single contrast mechanism. And we can get a picture of cells or blood vessels or whatever it is that we've labeled in that channel. But in our lab, what we have done is we've created or developed a multi-contrast system where one contrast mechanism gives us one variable. And then we use another contrast mechanism. In this case, it's called IOS, or intrinsic optical signal imaging, to get oxygenation. And another one called laser speckle contrast imaging to get blood flow. And this is what the images actually look like from these methods. So we have cells that are fluorescent, which allow us to identify the tumor extent in our models. So you can see here a bright green tumor in a live animal. We can image the same tumor with multiple wavelengths and do some very simple computations to get what's called the total hemoglobin or blood volume map. And we can get the intravascular oxygen saturation shown here. And then we can also look at the blood flow in the tumor using the same optical bench. So these are three completely different channels, not different wavelengths, but actually completely different contrast mechanisms that inform on three different physiological variables. Another thing that we do in our lab is we use orthotopic tumor models. So we take a tumor model, whether it's a brain tumor model or breast tumor model, and we inoculate it at the correct organ site. So for example, we put breast tumor cells in the mammary fat pad. We put brain tumor cells in the brain of the animal. And this is usually done so that we get tumor microenvironments that mimic what we see in patients. A lot of people do heterotopic tumors, as shown here. So these are basically breast tumor cells or any other type of cancer cells put in the dorsal skin flap of a mouse. And the reason people do that is because you can make the skin flap very thin and you can make it very rigid, so it's very nice for microscopy. So it doesn't move and it's easy to image. But we use something like this. So this is a dissection view of a breast tumor grown in the mammary fat pad of a mouse that's been euthanized. And you can see the breast tumor growing here very robustly. And then you can actually see angiogenesis, or the birth of new blood vessels. So blood vessels are recruited from the host tissue towards the tumor to support the proliferation and metastases of this implanted tumor. So it's a very good tumor model. So then Dr. Senaratna, who's a fellow in our lab, he and I developed this benchtop imaging system. And this is what the system looks like. This is a mouse that's been anesthetized. There's a little window here. I'll show you another picture of this soon. A couple of illumination sources, a camera, and a bed, if you will, to adjust the focus. So we implant breast cancer cells. We wait for a couple of weeks. We anesthetize the animals. We surgically expose the tumor. We implant a window, and then we image it. And this is what the window looks like. So we've actually 3D printed all these parts. I surgically expose the window, and then you can actually see the tumor and the best blood vessels underneath. Is we can actually image the blood flow, the blood volume, and the oxygen saturation in all these different regions at the scale of individual blood vessels. So this is a movie of the blood volume changes that occur inside a tumor over a 30-minute period. We often think of blood flow and blood volume as being very stable and very uniform. So shown here is the tumor. You can see very clearly the dilated tortuous vessels in a tumor. These are the collateral vessels in the mammary fat pad and the surrounding skin. And you'll see, let me see if I can run this. Yes, you can actually see how the blood flow, the blood volume, sorry, in each of these vessels kind of turns on and off. In some large vessels like here, it's very steady. But if you look inside the tumor, you get these little patches of darkness, because in tumors, the blood flow is very poorly regulated, and you get very abnormal flow and volume characteristics. From the same field of view, we can actually make maps of the oxygen saturation. So you can actually see in the normal skin, the blood vessels have very high oxygen saturation, but in the tumor, it's very low because the tumor is very poorly perfused. So once we have this kind of image, what we can do is we can zoom in on any area, and we can identify individual blood vessels. So on the left column here is just a small area from that entire field of view where we've identified a bunch of blood vessels. The top row maps blood flow, the middle row blood volume, the last row oxygen saturation. And what we can see in the first column, which is the mean value of each parameter in each vessel segment. So we can see blood flow is high in this vessel segment, low in the others. The the blood volume is also high here, and the oxygen saturation high here too. This is the variance in those parameters, and this is the power ratio of those two parameters. And then what we can do is we can look at correlations between these physiological variables. So we can see that in this large vessel, the blood flow and the blood volume are very well tightly coupled and correlated, as you would expect in a mature vessel or in a healthy tissue bed. But the rest of the blood vessels show very poor correlation. You can do the same thing for blood volume and oxygen saturation and blood flow. And as you can see, in general, in most tumor regions, the correlation tends to be poor, and then you can actually map the distribution of these correlation coefficients. So again, this is a very powerful toolbox that we call Henosis. We just published this. This toolbox has been made freely available to any investigators who want to use it. It's a MATLAB-based toolbox. You can go to our website and download it for your own applications if you have imaging data of this nature. The other thing that we can do is, instead of looking at just a small region of tumor, we can actually compute these parameters for the whole tumor. So we can, again, extract structural parameters of the vasculature. So this is microvessel density, the average vessel length, and the tortuosity over one whole tumor. We can map the average SO2, blood volume, and blood flow over the whole tumor, the variance and the power again. And we can make these really gorgeous maps. We can look at correlations between these structural and functional parameters, and we can see that some of them are really well correlated with each other, others not so much. And then we can actually do something quite clever, is we can actually take any combination of these morphological and physiological parameters and use them as basis functions to see if we can classify different regions of the tumor into unique niches, just like we did for that earlier data set that I showed you, based upon what we measure in the animal. So for example, using these 7 parameters, we were able to stratify this tumor into 3 distinct regions just based upon these characteristics. So again, depending on the tumor model and what you're interested in, this is a very powerful toolbox that leverages a whole slew of parameters to really interrogate the tumor microenvironment at the scale of individual microvessels. We can also look at heterogeneity in these tumors. So we can do this for 5 different tumors. So these are, as you can see here, from T1 to T5 are 5 different breast tumor models. And this just shows the images from these tumors over time from 0 to 60 minutes. And then we can actually extract these parameters for, these are the parameters average over all 5 tumors. These are the parameters for individual tumors. And one can immediately begin to see that, first of all, there are huge differences between tumors. So intra-tumor heterogeneity in these parameters, even though these tumors were all imaged at the same stage. And then there's also intra-tumor heterogeneity. So if I image a tumor at 1 hour and then at the second, image it again after 2 hours, you see very different characteristics in the flow profiles and the hemodynamics of these tumors. So again, a very nice toolbox to start looking at what's happening inside the tumor, especially if you're interested in identifying novel drug targets, or looking at the efficacy of drugs, or looking at drug distribution over time. Now I'm going to shift gears a little bit and show you how we've leveraged some of these tools in the tissue engineering space. So one of the things that we're working on with Dr. Warren Grayson's lab, who he's also a member of the INBT and a professor in the Department of Biomedical Engineering. So he's very interested in patients who have these craniomaxillofacial injuries. So the idea is to harvest bone for people who've lost bone in these areas and create implants and then encourage wound healing and new bone formation. And this is like almost fixing a filling in a jigsaw puzzle. And the idea is to somehow improve the outcomes for these patients, because when you harness or harvest a bone from, let's say, the leg of a patient or the hip of a patient, it is very difficult to get that bone geometry to conform to the wound site geometry. So we need a better method to actually treat these patients, because the wound site geometry is complicated. We often have variability in the sites of these injuries from patient to patient. So the idea is maybe to use some kind of biomaterial to help healing. But we can't just use any biomaterials, because some of them are not bioinert and they can have adverse side reactions. And also you don't want contamination at the trauma site. So what Warren is doing in his lab is he's actually creating these specialized bioactive scaffolds. So these are scaffolds that are made of specialized polymers that are biocompatible, they're biodegradable, they're mechanically durable, they can be customized to each patient, and they're very easily reproducible. And what he's doing is, from 3D imaging data, what we can do is we can 3D print the missing bone using the scaffold material. So it's shown here as a zoom version of the scaffold with these spores. It's made of a material called PCL. And then he impregnates this scaffold with a gel that's been loaded with adipose-derived stem cells and supplemented with some kind of growth factor, and then loaded into the scaffold. Because the idea is not just to have an inert kind of scaffold to encourage wound healing, but to have an active scaffold where you have stem cells that are capable of differentiating into new bone and new blood vessels. So one of the questions we set out to answer using our benchtop imaging system is, in an animal model of this injury, can we use this multi-contrast optical imaging pipeline to look at what happens in the tissue environment? So we can use, again, the same method, IOS or intrinsic optical signal imaging, to look at changes in vascular morphology in the bone graft region. We can use laser spectral imaging to look at how well this tissue or graft site is perfused. And then we can use fluorescence imaging to either look at something injected into the blood vessels or to track cells that have been injected into that area. So shown here are some very initial results from an early study we did with Warren. So this is an animal with what's called a calvarial defect model. So the skull has been injured, analogous to an injury that you'd see in a patient. And then that injury has been impregnated with a scaffold loaded with this fibrin gel and stem cells. So at week 1, through this window, you can actually see the scaffold. You can see some very weak vascularization in that region. And then you can see that the blood flow signal in that area is very, very low. There's almost no new blood vessels there. But if you wait for 2 weeks, what you see is you see new tissue forming around and within the scaffold. You also see new blood vessels coming in to nourish the growth of that tissue. And then you see blood flow occurring in these newly formed blood vessels. So this is very exciting to see, because now you have a tissue engineering construct, and the same benchtop system that informed the tumor microenvironment is now giving us invaluable information about the tissue microenvironment. We can then extract this vascular information. We can automatically segment it using some image processing tools, and we can actually characterize the vessel growth from week 1 to week 2 and changes in length from week 1 to week 2. We can also look at how mature these vessels are. So what we can do is we can have the animal breathe a certain gas mixture. In this case, the gas mixture used was carbogen. So we make the animal breathe either carbogen, which is basically 95% oxygen with 5% carbon dioxide. Carbon dioxide is a very potent vasodilator. So what we do is we turn this gas mixture on and off, on and off. And what we expect to see is we expect mature vessels to respond to this on-off inhalation of this gas mixture. So at week 1, since there's very poor vascularization, we see almost no response. At week 2, we see towards the periphery of the graft, really mature vessels that are capable of showing this response. You can see these 2 peaks. And then another part of the vasculature that shows you even a more robust response. We can get then pretty sophisticated about our image processing and identify different parts of the vessel structure by injecting a fluorescent dye. So we inject a dye into the animal, we allow the dye to transit our field of view, and then we can actually calculate at what time the dye arrives in different blood vessels. So shown here is the arrival time of the tracer, ranging from 1 second to almost 2 seconds. We can identify the time to peak within those blood vessels, and then we can identify the rise time of those blood vessels. And the reason we do this is because what you expect is the arterial or the fast-flowing vessels to have very quick arrival times, or the veins or vein type or vein-like vessels to have slower clearance times. We can also use that gas inhalation data to identify vessels that have responded very robustly or very poorly in that vessel bed to identify different vessel classes, ranging from very mature vessels to immature vessels, and then vessels that are a juxtaposition of those two phenotypes. So again, this is a very nice way to use imaging data to inform the design of bioactive scaffolds, so that with the end goal of translating these technologies into the clinic to help wound healing in patients with these cranio-maxillofacial injuries. So this is an ongoing area of work. The last few minutes I'm going to spend on some very cool technology we're developing in our lab to basically image the blood vessel changes that occur in the brains of awake animals. So traditionally, when we've wanted to image the brains of animals, animals need to be anesthetized or restrained. And we do that because when you're imaging something at very high spatial scales, you want to minimize motion so that there is no blurring of the images that we acquire. But when we anesthetize an animal or restrain an animal, two things happen. One, when we are trying to look at brain function, we cannot separate out the confounding effects of anesthetics on what we're measuring. And also, if the animal's restrained, we cannot correlate what we're measuring in the brain with the behavior of the animal. So if we're looking at some kind of behavioral phenotype, it's very tough to correlate the two. The other thing is, it's very hard to do these kinds of experiments if you're interested in the life cycle, or if you're interested in imaging the animal over the entire life cycle of the disease model. So say, for example, you're imaging an Alzheimer's animal or a brain tumor animal. It's very hard to do MRI on an animal every single day till the animal succumbs to the disease. And then, of course, a lot of these imaging methods like MRI, CT, especially in the preclinical space, tend to be very expensive. So like I said, we have a benchtop imaging system. This is what our benchtop imaging system looks like. We have a bunch of lasers, we have a nice camera, we have an imaging stage. And the idea was to shrink this imaging system down to a miniaturized system. So that's actually to scale. And we actually built this specialized microscope that mounts onto the head of an animal. So it's about 3 grams in weight. It's barely 5 centimeter cubes. So you can see how small this microscope is. And this is a quarter, a US quarter next to the microscope. Just to give you an appreciation of how miniaturized this new microscope is. And it does 3 kinds of imaging, just like the benchtop system I showed you earlier. It does fluorescence, it does IOS, and it does laser spectral imaging. So this little device actually gives us the exact same data that our entire benchtop system gives. This schematic shows us the actual structure of the microscope that we built. So this is the bottom view of the microscope. So we 3D printed these parts. Everything else is off the shelf. We have multiple light sources depending upon the type of imaging we want to do. We have a beam expander. We have a laser diode. We have some screws here to adjust the angle of the laser diode. We have a blue LED for IOS. For fluorescence, a green LED for IOS. And then we have a sync LED. So if you're doing some other kind of recording, you can actually sync it to what we're imaging. And then these parts actually fit onto a base with a lens and a couple of filters to remove the excitation light. And then the whole microscope actually snaps into a 3D printed frame that's glued onto the mouse's cranium. So shown here is a movie of a mouse that has been habituated to the microscope. So this is the microscope with some strain relief that's outside your field of view, and the animal's happily moving about. And then these are 3 movies taken from a freely moving animal. So the first movie, let me see if I can get this to play, shows you the transit of a fluorescent tracer through the field of view. So you can see the arteries light up, then the veins, as the tracer transits that vascular bed. The next shows how neurons light up when the animal is presented with a tone. So you see this flash of neurons averaged over multiple trials. Every time the animal is presented with a tone, you can actually see the neurons fire in that part of the auditory cortex. And then the last one actually shows you how the blood volume changes in response to that tone. So you can see you get this global increase in blood volume, which is very well known from the neuroscience literature, to supply those neurons with oxygenated blood. So you can see this is a very powerful tool to look at multiple things in live, freely behaving, unanesthetized animals. The real power of this microscope is actually apparent when you try to track a disease model over its entire lifetime. So what we have here is an animal inoculated with a brain tumor. The brain tumor cells are fluorescent. So everywhere you see bright green is where you see the presence of a tumor. So initially you see barely any green because there's no proliferation of tumor cells. But then as you go from day 3 to day 15, almost 2 weeks, you see the entire field of view becoming bright green because the tumor is actually growing and proliferating. This next row actually is a map of the cerebral blood volume. Again, there's no blood vessels in the region of the tumor. Then as the tumor grows, you can see these new blood vessels beginning to come in, indicated by the red vessels. These blood vessels come in to sustain the tumor growth. They anastomose and form connections, and then they kind of sustain the growth of the tumor all the way to the end. This is a map of the blood flow. You can see there's no blood vessels in the first few, for the first week or so. Then you see blood flow occurring in these newly formed vessels. And then you get really stabilized blood flow as indicated in these later images that sustain the growth of the tumor. Again, you need to bear in mind that this is an awake, freely behaving animal taken over the entire lifetime of the tumor. The other thing that we did for this microscope is we built like a very simple 3D printed adapter that allows us to take the microscope's magnification from 5 microns to 0.5 microns. So this is what the blood vessel architecture looks like without the adapter. With the adapter, you can actually zoom in and look at really fine vessel structure. And if you inject a fluorescent dye, you can actually make movies of this nature where you can actually see erythrocytes or red blood cells trafficking through these little capillaries. So extremely high resolution imaging in a live animal. And then once we have movies of that nature, we can use some image processing tools and tricks to identify the erythrocytes over these different imaging frames. And then we can make maps like this where we can plot the blood flow in each vessel segment as a function of the erythrocytes' motion over time. So I think that's it for today. And what I'd like you to do is, again, please check out our lab's website, because we have a lot of information there regarding our current work and some of the ongoing papers that we have, or papers we've presented, and a lot of the protocols that we use in these studies. I'd like to thank Dr. Popel, Grayson, and Thekor, all in BME, who are very close collaborators, Dr. Tyler in neurosurgery, who worked with us on the brain tumor work, and the Division of Cancer Imaging Research, where I am located in the School of Medicine. Of course, I'm the messenger. I'm very fortunate to have an incredible group of postdocs, graduate students, and undergraduate students working in the lab. So if you're interested in this kind of work, again, please hit us up or shoot me an email. And I'd like to thank our funding sources, which made this work possible. So thank you for your attention, and I'm happy to take any questions you might have.
And thank you very much, Dr. Pathak. If anybody has any questions, please feel free to use the Q&A feature at the bottom of your screen.
Well, I guess it was all very clear.
Any questions for Dr. Pathak? Okay, we have one here. Can you see this?
Let me see. Yes. I was just wondering, how can you resolve hemoglobin and oxygen saturation so effectively in your tumor imaging instances? Okay, so one, there are two factors bundled into your question. The first is that of spatial resolution. So the spatial resolution of the benchtop system is actually quite high. It's about 5 microns. So you can actually resolve the blood vessels. But the hemoglobin oxygenation versus deoxyhemoglobin can be resolved by using multiple wavelengths to excite those blood vessels. So we use multiple wavelengths. We have a white light source and a filter wheel. We illuminate the blood vessel using multiple wavelengths. And then we use the reflectance images in a model that allows us to calculate what the oxygen saturation is in those blood vessels, from which we can then extract those parameters.
Thank you for your question. Does anybody else have any questions? We have a question from Eugenia.
Okay, let me just take a quick look at it. Have you examined, thank you for your question and your comment, Eugenia. Have you examined whether there are differences in intra and inter-tumor heterogeneity among different tumor types? Our breast scans, oh, great question. No, we haven't explicitly or systematically looked at differences, but other people have. And just from our work in general, I can tell you, yes, there are dramatic differences in the blood vessels of different tumor models, organ systems. So the answer to your question is, yes, there are differences. Have I looked at them systematically? The answer is no, other people have. We have a question from Jianqing. Do you enjoy the arts? Yes, very much so. Everything I do is informed by my love for the arts. In fact, I see Lydia Gregg on this call. We're actually collaborating with Lydia, who's a professor in the Artists Applied to Medicine program. And we're co-mentoring a graduate student in that program to actually leverage some artistic and visualization tools to make our data communicate these points even more effectively. So yes, I do enjoy the arts, and thank you for your question.
If anybody has any additional questions, feel free to type them now. Okay, well, I think that concludes our seminar for today. Thank you so much, Dr. Pathak, for coming and speaking with us. We really appreciate having you here.
Thank you so much for having me. I hope everybody enjoyed that. Have a good afternoon.
Thank you.
Bye-bye.
Thank you, everyone.
Thank you for coming, everyone.